Hello everyone, my name is Dimitri Perrin and I want to talk to you about some work we've done as part of the CAMDA 2020 Metagenomic Geolocation Challenge, and in particular the approach that we've taken using read signatures. This is work carried out at the Queensland University of Technology with my colleague Timothy Chappell, Shlomo Geva, Jim Hogan and David Lovell. Today I will start by giving you an overview of the data we were working with and the approach we took for this challenge. So the data for the CAMDA 2020 Geolocation Challenge is a subset of the data collected as part of the MetaSub consortium. So that consortium overall now has over 4,000 samples in total, but as part of the challenge we were given 1,065 samples from two collections, the 2016 and 2017 collections. These samples represent 23 different cities. For each city, we have between 19 and 99 samples, with a median of 45 samples per city. In each sample, we had from 160,000 to 482 million reads. So quite a big spread in terms of the size of each file. The median number of reads per sample was about 4 million. These reads are paired end reads, and so that's our initial data. As a bonus, we also have some mystery samples for which the origin is unknown to us, at least in the first instance. And so that's the data that we've been using for this challenge. And when we approached the challenge, the approach that we took was to develop a method that was going to be scalable, interpretable, and customizable. So the reason why we want something that is going to be scalable is that we're working with a very large number of samples. We're working with 1,000 samples, but the entire MetaSub collection, if we ever want to look at the entire collection, is even larger. It already has more than 4,000 files. And so it's very important to develop a method that is going to be able to scale to these very large collections. What we mean by interpretable is that we want a method where for each step we're able to understand and reflect on the behavior of the method so that we can see what works, what doesn't work, and what we might be able to improve. And the only way we can do that efficiently is if we're not working with a black box. And what we mean by customizable is that we want a method that can easily be modified or refined as we understand more about its behavior. So with these objectives in mind, what did our approach look like? Well, at a very high level, we have 4 main steps. In the first step, we preprocess the reads. So for each sample, we work with each sample individually, and we just preprocess the reads to prepare them for the analysis. Then we use a representation of each of these reads using signatures. We cluster these signatures to represent a sample, and then using these clustering results, we classify samples. So what I will do now is go through each of these 4 steps and give you more details about how we approach them. So, as I mentioned, the first step is the preprocessing. Our starting point is the Illumina paired end reads from these 1,065 location samples. When we tried to access the files, some of the compressed files were partially corrupted, but we still managed to extract almost 9 billion pairs from these samples. They're paired end reads, so we used pair to merge all these pairs, and we successfully merged about a third of the pairs, and so we end up with 3 billion sequences. And that's the sequences that we use for the analysis. One thing we could do later on is explore what happens with what we could do with the pairs that we could not merge, but in these analyses that I'm discussing today, we focused on the reads that we were able to merge. Now that we have our merge reads, the first thing we did was to try to eliminate reads of human origin. The reason why we did that is because we think that given the context of the acquisition of the data, given that it's the metasub data, it's public transport, so we expect that there will be humans for each of these environments, and so we think that the human reads are not going to be very informative in terms of location. So we wanted to explore how we could get better results by removing these reads. So the way we've done that is with Botai, with the sensitive local settings, and using the reference genome as well as the major SNPs pre-built as in the index. And so we run all of our 3 billion merged reads through this Botai process. And what we found is that overall, about 15% of the collection was mapped to the human genome. And so the remaining 2.4 billion unmapped sequences moved to the next step of the process. If we look at individual samples, there's quite a bit of variation. We have some samples where we have virtually no human reads, so we only mapped 0.1% of the reads to the human genome, but we also have samples, at the other hand, where we mapped 97.2% of the reads to the human genome. We looked at the distribution of these alignment rates to the human genome for different cities. And you can see that from this a spread, but for most cities, we have some samples with no human reads and some samples with a high number of human reads. Two exceptions are Ofa and Lisbon on the left-hand side, where we virtually have no human reads in any of the samples. When we submitted this analysis, one of the reviewers was supportive of this idea of filtering human reads, but also suggested that, well, if we're going to remove reads of human origin because they're not informative, we might also want to remove bacterial reads that also have a human origin because they're species that are part of the normal skin microbiome. And so that reviewer suggested that we also try to remove reads from the Cutibacterium acnes species. So we took that idea on board and we repeated the same process of filtering using Botai, using the representative genome for that species. What we found is that approximately 1.4% of the sequences that were not mapped to the human genome were mapped to the C. acnes genome. And so same thing as before, we use the unmapped sequences for the rest of the analysis. And same pattern as for the human reads, there's a big spread. We found some samples that did not contain C. acnes and some samples that had a very, very high proportion of C. acnes reads. And so we did the same thing as for the human genome. We looked at the distribution in different cities. And again, you can see that Lisbon and Ofa are outliers where nothing is mapping. But then in most other cities, we have some samples that have very low C. acnes content and some samples that have very high C. acnes content. And in a way, like these results on the human reads and on the C. acnes reads validates our idea that these reads are not informative because they're present in most cities, but within each city there's quite a big variation, and so the presence or absence of such a read is not really indicative of the location. It's only indicative of the city where that sample is coming from. Again, so that's what we did for the preprocessing. Paired merging, and then 2 steps of filtering to remove reads we think are not informative. Next, we can move to our second step in the process, which is the representation of the data. What we're doing here is that we want to move from reads to some representation. And so the reads that we're working with, so the initial paired end reads are 151 bases in length, but as part of the merging process, that the length of the overlap can vary, and so we end up with merged reads that have a length roughly from 50 to 292 bases. And so what we do is that as a first step, we decompose each read into its constituent k-mers. And then for each of these k-mers, we use a lookup table to encode each of the read k-mers as a fixed length vector. Okay, so we have an encoding of all the canonical k-mers, and so for each read, each k-mer that is present in the read, we can go and look at that fixed length representation for the vector, for the k-mer. Here in that figure, we're showing an encoding that's based on random unit vectors, but there are different strategies there, and I will come back to this later. Once we've identified the encoding for each of the k-mers of a given read, we sum these reads and we normalize them to a vector of unit length, and that's what we call the signature for the read. Okay, so to summarize, we have a read, we extract the k-mers from this read, we look them up, we sum all of the signatures from individual k-mers, and that we normalize it, and that gives us the signature for the read. Now that we have these signatures, we can more efficiently calculate distances. We don't have to use string operations, so we can perform clustering much faster, and that's the next step of our analysis. Okay, so we want to go from signatures for individual reads to signatures for locations. So what we did is that we generated read signatures using the method I've just shown for up to 1 million reads per location. That 1 million limit is not something that has to stay like this. It's just like a temporary value we used just to ensure that we could repeat complete enough tests in time for the submission. There's nothing that's forcing the method to stop at 1 million reads per location. We can increase that later on. But with that 1 million read per location limit, we generated just under 1 billion signatures in total. And what we want is that we want a more compact representation for each location. And so what we're doing is that for each location individually, we cluster the reads using these signatures. And the advantage of doing that is that a location can now be represented by the signatures of the centroids of the clusters we've generated for that location. Okay, so if we have M clusters for a location, we're going to move from 1 million signatures for all the reads for that location to just M signatures, one for each of the clusters that we generated. So how did we cluster? Well, for each location, we took the read signatures that we generated and we clustered them using TSVQ as the clustering algorithm and the Euclidean distance. TSVQ is a hierarchical top-down algorithm based on k-means. Here, we use k is equal to 10, so we first generated 10 clusters, and then for each of these clusters, we run k-means again and generate 10 subclusters. Okay, so that means that we end up with overall 100 clusters by just running k-means with k is equal to 10 11 times. And this means that for each location, we can now represent that location using just one signature, which would be the overall centroid, or 10 signatures, 1 per centroid of the 10 clusters, or 100 signatures if we use the centroids of all 100 subclusters. And in the next few slides, I will show you the results for these three approaches: 1 signature, 10 signatures, or 100 signatures. Before we get to that, the last step of the process is the classification. So to classify any sample S, what we do is that we take up to 1 million read sequences from S, we encode them using our signatures, then we cluster them using TSVQ, and we obtain some clusters. And for each of these clusters, we find the nearest neighbor in our reference collection. And then we predict the location of each of these mystery clusters from S as that of that nearest neighbor in the labeled collection. And then we accumulate the cluster predictions. So if we're working with just one cluster per location, well, we have nothing to accumulate. But if we're working with 10 clusters or 100 clusters, well, we have 10 or 100 predictions. And so we just accumulate these predictions, and the final classification is for sample S is going to be the city that is associated with the location having the most clusters appearing as nearest neighbors of clusters from S. Okay, so a simple voting mechanism. Let's look at some of the results we obtained using this approach. As an initial validation, we used leave one out cross-validation to look at the performance of this classifier. So we use all 1065 locations. And what we do is that each location sample is left out in turn. So we remove one sample, and we try to predict the city of that sample using the remaining 1064 samples. And we calculate the accuracy of the classifier as the number of correct predictions over the total number of predictions that we're making. Okay, let's look at the results we get running the cross-validation over these 1065 samples. So here I'm showing you the results that we obtained using formers as the k-mer length, using just one cluster, and using the nearest neighbor for the classification. And so I'm showing you the confusion matrix. So we have all the different cities, and so on the diagonal we have the correct classifications. And then in the confusion matrix, I'll also show you things that have been mislabeled. Okay, so the first message is from these results with one cluster is that we're doing much better than we would expect on average. And as you would expect, these results are going to get better as we use more clusters. So if we use 10 clusters instead of just one cluster, and still use the nearest neighbor for each cluster, and then a vote for the final classification, we can see that these results are improved, and so most samples are correctly assigned to their city of origin. And if we go to 100 clusters, the results are further improved. Okay, so here, still using formers, now we use all 100 clusters from TSVQ. For each of the clusters, we find the nearest neighbor, we vote, and we use the results of that vote for classification. And we can see that for most cities on the diagonal, we have a correct classification. There are a couple of cities that are worth highlighting. The first one is Lisbon, where we don't get any correct classification. If you remember the results on the filtering, we also could not find any human reads in Lisbon or any Cutibacterium acnes reads. So we're not sure exactly what is going on with Lisbon. It's something we want to explore further later on, but we're having trouble with these samples. The other thing that's interesting to look at is the city of Offa, where we only correctly classify 7 samples, and 19 are misclassified as coming from the city of Ilorin. So that was a little bit surprising at first, but what we realized is that Ilorin and Offa are 2 cities that are only 40 kilometers apart in Nigeria. And so we could argue that, well, it is actually just one location. Some of these samples might be, even though they're labeled as different cities, they might actually be closer to each other than 2 samples from Tokyo, for instance. And so that these 19 misclassifications of Offa as Ilorin could arguably be counted as correct classifications. And so here it's an extreme case because the 2 cities are only 40 kilometers apart, but we can see that there are some other cities where there might be a geographical component to the misclassification. So for instance, Denver, most of the misclassification is in New York. Tokyo, we're doing well overall, but the most abundant, the most frequent misclassification is Sendai, which is also in Japan. We have something similar with reads from cities in Asia, sometimes being misclassified as coming from Kuala Lumpur. So for instance, for Hong Kong, we get 15 right, and 6 of the ones we're getting wrong are coming from Kuala Lumpur, and 7 are coming from Singapore. So it gives us some confidence about what our method is doing. Then, taking advantage of the fact that our method is an open box and we can look at what each source that we've made is doing, we try to investigate the impact of some of the decisions and some of the parameters that we're using. So the first thing we looked at is the influence of the initial filtering. And so we looked at the performance with the reads that we obtained directly after the merging, or if we remove the human reads, or if we also remove the Cutibacterium acnes reads. And as you can see, there is a performance boost, so we get more accurate as we remove reads that we assume not to be informative. The biggest jump is when we remove the human reads, but we still get a small performance boost by removing the C. acnes reads. One thing to keep in mind is that here we have a 23 class problem. We have 23 cities, and so if we were randomly guessing where a sample is coming from, we would expect to be correct about 4% of the time. So an accuracy of 40 to 65% is actually quite good. Another thing to keep in mind here is that if we have a tie in the voting process, we do not count that as a correct classification. So for instance, if we have a sample from Tokyo and we say we have 5 votes for Tokyo and 5 votes for Sendai, we do not count that as correctly classifying it as a Tokyo sample. We also looked at the length of the k-mers and whether that had an impact. Because you would expect that longer k-mers carry more information, and so as we can see here, that's the case. And as we increase the length of the k-mer, we get more accurate. The interesting thing to note is that even for dinucleotides, even for tumors, we still get fairly good results, which we did not expect at all. The results I'm showing here are the ones we obtained after removing the human and C. acnes reads. The advantage, of course, of using shorter k-mers is that the overall method is going to be faster. So there's always a trade-off between using longer k-mers that will have a higher accuracy and shorter k-mers that will be faster to compute. The next thing we wanted to look at is whether there was the number of samples we had for each city had an impact on the classification. If you remember one of the first slides when I explained the data set, we have between 19 and 99 samples for each city, and so we wanted to make sure that that didn't introduce a bias. And so if on this graph, we're plotting the classification accuracy as a function of the number of samples we have for a city. And as you can see, there's no clear trend. So we're not necessarily doing much better for larger classes. We're also, on that graph, showing the processing labs as the color of the dots. So the red dots are samples that have been processed in New York City, blue dots have been processed in Shanghai, and green dots have been processed in Stockholm. And so we just wanted to confirm that there was also no lab effect in the results of our methods. And yeah, that's what we observed here. Just a quick note on these processing labs. That's our best guess as to where they've been processed. That information was not provided as part of the CAMDA challenge, but it's something that's mentioned on the Metasub website. They say that they use only these 3 processing centers, and so we manually assigned the locations to a processing lab. I mentioned a couple of slides ago that there are cases where the voting step results in a tie between n cities, often between 2 cities, but you could have cases where you have a tie between 3 or 4 cities. In the previous results that I've shown, we counted it as an incorrect classification, even if the correct location is actually included in the tie. Well, an alternative would be to accept it as a correct classification. So for instance, we could accept as a correct classification a tie between 2 cities as long as the correct location is one of the 2 cities in the tie. Well, if we do that, we do get a slight increase in the accuracy, but it doesn't make a big difference. And as we go to longer k-mers and higher numbers of clusters, that difference become negligible. It's only 1 percentage point, for instance, if we use 4 mers in 100 clusters. Another important thing to consider, of course, is how long the method takes overall. So what I'm showing on this slide is the timing for each of the steps. And so you can see that the pre-processing, it takes some time. So the paired end merging takes about 15 hours for the entire collection. And the read filtering takes about a day and a half over the entire collection of 165 samples for each species that we want to filter. But once we've done that, have that pre-processing out of the way, the actual method is very fast. So the signature creation, clustering, and classification is extremely fast. Even with 5 mers, which gave us a very high accuracy, it takes less than 8 hours end to end to run that entire process. And if we move to shorter k-mers, we can even manage to run the whole pipeline in less than an hour. The last result I want to touch on is the mystery samples that I mentioned at the very beginning. So for these mystery samples, now that we've seen the labels after a submission, there are 2 categories of cities. There are some cities that we revisit, so cities we've seen in the 1065 samples, and some new ones. So for these revisited cities, if we use 5 mers, 100 clusters, and we remove the human and Ciacness rates as before, we get an accuracy of about 44%, which is similar to what we got for those 5 cities in the cross-validation, where we got about 49% accuracy for these ones. And so it confirms that our approach is robust. For new cities, of course, our approach is always wrong because the correct location doesn't exist in our classifier. But what we did is looked at how they were classified, and we saw that the classification that we attempted is not necessarily a neighboring city. And so, for instance, we see that many of them are classified by default as coming from New York, which is not surprising given that New York is the largest class. It has with 99 samples. What I would like to do in the last few minutes is just summarize the approach and discuss a little bit where we think the next steps might be. So our approach that we've developed for this challenge is to rely on short k-mers, signature encoding, and clustering to identify the location of a sample. And this method proved to be quite fast, taking only about 2 days, including the preprocessing. And that's quite fast given that the entire size of the collection is almost 9 billion reads, and most of the time is actually spent on the preprocessing, which we can optimize further. And if we look at just at the signature encoding, the clustering, and the classification, that takes less than 8 hours, even working with 100 clusters and 5 mers. The method is also interpretable, as it does not involve any black box at any step. It means we can look at the impact of each decision we make along the way and further refined based on that, because our method is completely customizable. And I want to give you an idea of the next few things we want to try on that front. One of the first things we want to look at is to revisit the k-mer projection methods. Here we used an orthogonal basis that had the same dimension as the number of canonical k-mers. But there are other things we could do. We could do some projections, we could do some dimensionality reduction, we could use binary signatures instead of real value signatures. All of these would give us a speed advantage, and so we want to further explore these different projection methods. We also want to look at different distance metrics. Here we use the Euclidean distance as a starting point, but we know that in high dimensions it can be problematic. So we want to look at other metrics that we can consider, such as fractional Minkowski distances. We also want to look further into the different clustering algorithms. Here we use TSVQ to create the 100 clusters, but that's taking a shortcut. We create 10 clusters, so using k-means with 10, and then for each of these we create 10 subclusters, which is again using k-means with k equal to 10. So they're just running k-means 11 times, which is faster, of course, than running k-means with 100 clusters directly, but doing that would be more accurate. So we want to explore that a bit more and maybe consider other clustering algorithms as well. The next thing we want to look at is the classification. In this study, we use the nearest neighbor only when classifying a specific cluster, but we could very easily extend that and use the k nearest neighbors instead. And finally, the last thing we want to look at is how we evaluate the performance. When we have ties, any way we break them is always somewhat arbitrary. And so we want to look at other ways to evaluate the quality of the classification. In particular, we're interested in the top n accuracy, which is to consider that a classification is correct if the actual location is present as one of the top n votes. And with that, I just want to thank you for listening. And if you want to know more about our approach, please get in touch. Thank you.